Welcome back, everyone, to this explainer. If you are gearing up for those brutal cognitive neuroscience exams, well, you've landed in exactly the right place. Today, we're taking all that complex neurobiology and turning it into your ultimate high-yield study guide on the cellular bases of memory. So let's put down the dense textbooks for just a minute. We're going to visually map out exactly how your brain physically changes to learn new information. Okay, here is our game plan for today. We'll start at the micro level with cellular mechanisms and synaptic plasticity, zoom out to multiple memory systems, look at amnesia and brain damage, and wrap it all up with neuroimaging.
Section 1, Cellular Mechanisms of Memory. We're starting right at the synapse.
So here's the foundational biological problem you're absolutely guaranteed to see on your test. How does the activity of different brain regions actually physically change to form a memory? Well, most models point to changes in the strength of synaptic interactions among neurons. Enter Hebbian learning. Formulated by the Canadian psychologist Donald Hebb way back in 1949, his rule basically states that if a synapse between two neurons is repeatedly activated at about the same time the postsynaptic neuron fires, the actual structure or chemistry of the neuron changes, and that connection gets stronger. Think of it kind of like walking through the woods. That first time you push through the brush, super difficult. But if you walk that exact same path repeatedly, simultaneous with your destination, you trample down the weeds, right? It becomes a clear, permanent trail. Now, if we take Hebb's concept just a step further, we hit a much more complex, general mechanism known as long-term potentiation, or LTP. When neural circuits, specifically in the hippocampus, are subjected to repeated, intense electrical stimulation, those cells become incredibly sensitive to stimuli. And that enhanced response? It can last for weeks, sometimes even longer. If Hebbian learning is walking that path in the woods, LTP is the biological crew that comes in and paves the trail with asphalt. As researchers like Badley suggest, this is literally the crucial mechanism for long-term learning and retention. Section 2, Chemistry of Synaptic Plasticity. Let's look at the molecular triggers. We know the trail gets paved, but we really need to know the specific neurochemistry that makes LTP possible. Definitely star this in your notes, because understanding these molecular triggers is a vital checkpoint for your upcoming exams. It all kicks off with glutamate. Glutamate is the major excitatory transmitter in the hippocampus. Think of it as the primary chemical key needed to unlock memory formation at the dendritic spines of postsynaptic neurons. And this key is capable of binding with two very specific types of locks, NMDA receptors and non-NMDA receptors. Now, you have to know the contrast between these two receptors. NMDA receptors are doubly gated. They're the ones central to producing long-term potentiation. Non-NMDA receptors, on the other hand, are central to maintaining it. To wrap your head around that doubly gated NMDA receptor, just imagine a double-locked door. To open it up, you need both a key, which is the glutamate, and you need a physical push, which is the postsynaptic depolarization. Only when you have both the excitatory input and the depolarization does that NMDA door fly open to actually produce LTP. So how do researchers actually prove this? Well, through chemical blocker experiments. When scientists introduce a drug called AP5 into the brains of normal mice, it specifically blocks those NMDA receptors. The locks are jammed, which prevents the induction of new LTP, which in turn completely impairs their ability to demonstrate normal place learning. Essentially, they lose their spatial memory. But, and here's the exam-winning detail, AP5 does not affect previously established LTP. It only stops the paving of new trails. This proves for sure that NMDA is for production, not maintenance. Moving on to Section 3, Multiple Memory Systems. We're zooming out to the cognitive level now. Shifting our narrative from the cellular chemistry up to broader cognitive systems, cognitive theory and neuroscientific evidence strongly argue that memory is not just one single monolithic process. Instead, it's supported by multiple, totally distinct subsystems. We're talking sensory registration, perceptual representation, all the way up through working, procedural, semantic, and episodic memory. The brain structures that support these processes differ entirely depending on the type of information you're retaining and exactly how it is being encoded and retrieved. For the exam, make sure you can distinctly contrast these systems, particularly episodic versus semantic memory. The distinctions in quality here are stark and readily identified. Episodic memory is your personal record of events and experiences, like remembering your very first day of college. Semantic memory, though, is your internal encyclopedia of facts and concepts, like knowing the chemical formula for water. They're encoded differently, retrieved differently, and rely on completely different neural networks.
Section 4, Amnesia and Brain Damage. Let's get into some clinical insights.
What happens when these carefully balanced biological and cognitive systems break down due to localized brain damage? Let's use a high-stakes clinical checkpoint straight from the source material, patient HM. With severe damage to his medial temporal lobe, this patient developed profound amnesia. So you gotta ask yourself, what specific form of amnesia did he develop? What information could he still retain? And critically, what exactly do findings from amnesic patients tell us about how memory operates and encodes in healthy people? Case studies like this are huge because they bridge the gap between theory and reality, showing us exactly how our healthy memory is compartmentalized.
Finally, Section 5, Neuroimaging and Future Research, Mapping the Mind.
Despite all the really intriguing results from neuropsychological studies of localized brain damage, a big question remains. Are memory aspects localized in one place or distributed across different cortical regions? To answer that, we turn to modern neuroscience, beautifully summarized here by Endel Talving. Neuroscientists today reject the idea of studying memory as though it were a single process. So instead of looking for a mythical general memory center, researchers are using advanced imaging to find the neurological underpinnings at a highly precise, isolated level, like mapping the exact networks for just encoding or just retrieval. So I'm going to challenge you with a Socratic question directly from your study materials. How do we actually design an experiment to study the neural process of semantic memory using functional MRI technology? Well, to isolate the retrieval of facts and concepts, you would design an experiment where a patient inside the scanner is asked to name objects or answer specific factual questions. Then, and here's the trick, you subtract the baseline brain activity of them simply looking at abstract shapes. This isolation of retrieval networks is exactly how the latest neuroimaging techniques will keep giving us invaluable info about healthy human memory in the years to come. To wrap things up today, I really want to leave you with a profound thought exercise. Just imagine what it would be like to actually recover from one of the forms of profound amnesia we've discussed. Try to describe your impressions of and your reactions to your newly recovered memory abilities. When we study NMDA receptors and temporal lobes, it is so easy to get lost in the chemistry, but this really reminds us of the immense human impact of these complex neural networks. Thank you all for joining me for this explainer. Keep visualizing those pathways, and seriously, best of luck crushing your exams.